This is Research Like a Pro, episode 244, RLP with DNA 7, Locality and Ethnicity. Welcome to Research Like a Pro, a genealogy podcast about taking your research to the next level, hosted by Nicole Dyer and Diana Elder, accredited genealogist professional. Diana and Nicole are the mother-daughter team at FamilyLocket.com and the authors of Research Like a Pro, a genealogist guide. With Robin Worthland, they also co-authored the companion volume, Research Like a Pro with DNA. Join Diana and Nicole as they discuss how to stay organized, make progress in their research, and solve difficult cases. Let's go! Today's episode is sponsored by newspapers.com. Break down genealogy brick walls with a subscription to the largest online newspaper archive. Hi everyone, welcome to Research Like a Pro.
Hi, Nicole. How are you today?
I am fabulous. How have things been going? What have you been working on?
Well, we just finished up Roots Tech, so I've been working on catching up after those few days of intense giving presentations and meeting people at our booth, but it was so great. So fun to meet so many of our listeners, and it was great to meet new people who have never listened to a podcast and don't know anything about it. It was fun explaining how it worked, and hopefully some of our new listeners will figure out how to do podcasts because it really is a great way to learn.
Yeah, it was fun to me how many people said, I listen to your podcast while I'm walking my dog.
Yeah. Well, and I love podcasts. I love listening to audiobooks. It just seems like a good way to do two things at once, listen and get some other tasks done. So I'm looking forward to my garden being able to come to life. We've had winter for so long, and I like to listen to podcasts while I'm gardening. So I'm hoping for some warmer weather someday.
Oh, true. You know, it's spring here in Tucson already, and our nectarine tree is out blossoming and bees buzzing all around it.
Oh man, that sounds so fun.
The Rooster Conference was so fun. I just wasn't sure what it would be like after 2 years of virtual due to COVID. And we wondered if it would be as big as it was in years past. And it was definitely smaller, but I thought it was a good amount of people. And I think in 2020, it was so crowded. The hallways were so stuffed with people that it was hard to get around. And this time, it was a perfect amount of people, in my opinion.
I agree. It felt really good. And our classrooms had plenty of room for everyone to spread out a little bit, but they still felt pretty full. So, especially the class that we did together, that one actually was really full. So, that was just fun to be in person and see people reacting to your talk. I don't mind doing webinars and video presentations as well through Zoom, but it's fun to see faces in person.
Right. And we decided just to do in-person talks this time and not pre-record any of our videos. So if you didn't attend in person, our lectures were not recorded. However, you can always watch us talking on our webinar series, and we would love to have you join that.
Right, we have got another fun new announcement, and that is a new Airtable quick reference guide that Nicole designed. So if you've been working with Airtable and you would love to have a guide to refer to help you with all the different features of Airtable, that's available as a PDF download on Family Locket in our shop. And then our Research Like a Pro webinar series for 2023 is well underway. We have monthly case studies featuring the Research Like a Pro and Research Like a Pro with DNA process. And so we invite you to join us for that. Those are all recorded, and whether you attend live or not, you have access to the series on Family Locket. And we have our next study group beginning this fall, very end of August. It'll go through November of 2023, and that will focus on purely documentary works. We also have a peer group leader application on our website if you'd like to join us as a peer group leader and have a complimentary registration. And as always, join our newsletter for coupons for special sales that we have going on. And the next big conference coming up is the National Genealogical Society Conference. That will be the very end of May, 1st of June in Richmond, Virginia. It will be an in-person conference with 10 lectures being part of their online presence, and that is now open for registration. And then there's also the Association of Professional Genealogists Professional Management Conference coming up in the fall, and they are having a call for presentations. So if you're a professional genealogist or you have something interesting you'd like to share with that group, you might want to check out that conference and submit a proposal.
So many fun things coming up. Our topic for today is Chapter 7 from our book, Research Like a Pro with DNA. And Chapter 7 is all about locality research and ethnicity. And when we were preparing the plan for Research Like a Pro with DNA, we were trying to figure out, what part of DNA evidence would we talk about in the locality research chapter? And we finally settled on ethnicity because a lot of what ethnicity is about is determining where your DNA is pointing to as far as location. And originally, the locations in our admixture were just very broad, but over time, they've developed several new ways to make it more specific, like communities and more specific locations within the more broad continents and countries. So it's exciting. So, usually, when you receive your DNA test results, that's the first thing to explore is that pie chart or ethnicity estimate to see what percentage of each ethnicity you are. And you wanted to match that up and see if it looks like the same as what your family tree looks like. Over time, you may have seen that there are updates to the ethnicity estimates. So it's important to understand what's going on here with these ethnicity estimates and really know what it can and can't tell us, because ethnicity estimates are estimates. They can play a role in our research, but they also can't tell us everything. And so our DNA does hold clues to where our ancestors came from and where they settled. So we shouldn't ignore this potential source of information, but we do need to analyze our ethnicity estimates and compare and contrast them across the various testing companies. So they can be useful when used in conjunction with documentary research, and often they can help us confirm or reject a hypothesis. Another way we can use them is when we're working to identify the most recent common ancestor with a DNA match. And looking at the locations and suggested ethnicities can help us reach that goal. Sometimes we can even reach out to our DNA matches and find out that they're an expert in a locality that might be able to help us. And they can share research information or access records and repositories and history of that area. So often our DNA matches are a resource to us to help us with our research and understanding localities that are far from us. So it's important to add locality and ethnicity information to our DNA research log to keep all of this information organized that we're learning. So in the previous chapter, we learned how to analyze our sources and make a timeline, and that timeline included specific locations. And we also evaluated our DNA matches and started looking at their trees and maybe started noticing some shared localities. So, to get ready for the next step, which is creating our research plan, we're going to do locality research. And that's going to require us to select a locality and prepare a locality guide to help us really dive into the history, geography, records, and repository that will inform our research path. So, this chapter is all about that. It's about weaving together locations in our matches trees, ethnicity results, and locality research.
Thank you for that introduction to this chapter. Let's begin by talking about the terms admixture and ethnicity estimates. You will hear both of those as you listen to lectures or read articles, and both of them refer to the same thing. We're talking about this genetic mix of various populations that make up our unique DNA. And so I'm just going to use the term ethnicity estimate because it's just easier to have one term, but no, when you hear admixture, that has the same general meaning. So let's just use my ethnicity as an example. My ancestry is broadly European from the British Isles, Western Europe, and Scandinavia. Well, as you know, those populations mix widely with one another. So at this point, the DNA testing companies just can give me broad estimates. But if you have a very diverse population in your background, such as Northwestern European and Mexican ancestry, you would have more success in distinguishing which portions of your ethnicity admixture originates from specific branches of your family tree. Here's a fun example of an individual's known ancestry of Latin America, British Isles, and Scandinavian. A pretty big difference. So the specific communities listed in Eastern Norway and Indigenous Americas / Mexico are useful clues for continuing research in those locations. So this person has 46% Norway with specific communities noted there, and then 11% England, Wales, and Northwestern Europe, 10% Ireland, 7% Scotland, then 7% Indigenous Americas / Mexico, then 6% Sweden, 5% Wales, 4% Spain, 2% Portugal, 1% Indigenous Americas Andean, and 1% Eastern Europe and Russia. That's a much more interesting set of ethnicities than I have in mind, which is pretty much just British Isles, Ireland, Scotland, and some Scandinavian. So it's so interesting to look at different people and see what their DNA shows. Now, you may have your DNA at several different companies, and you'll notice that each company defines geographic areas differently. One may include Denmark in a Scandinavian region, while another company has it in a broadly northern European region. And this is just because every company defines regions differently. Our ancestors migrated, and the ethnicity estimates are most accurate on the continental level. So some populations were more isolated, such as Finnish, Japanese, Ashkenazi Jews, and so those ethnicity estimates may be more accurate as a result. And with every update to the testing company's ethnicity estimates, I think we're able to see those become more narrowed down and a little bit more accurate. And it will be fun to see how things change as the science evolves. So how do these testing companies come up with these estimates? Well, each uses a proprietary algorithm to compare our DNA to that of reference populations worldwide. And that just means a group of people with known origins in a specific location. And because each testing company does use a different reference population, our ethnicity estimates vary among the websites. And they are continually adding to the reference populations and refining their science. So our ethnicity estimates will continue to change. We're going to take a look at each of the 5 main DNA testing companies and what they offer in the way of understanding our genetic origins. And we'll use my DNA estimates to illustrate the differences. And it's always just really fun to do the comparison. I also want to note that if you take screenshots of your ethnicity so you can compare through the years, that's another fun thing to do. So looking at my genealogical family tree, here is the breakdown. On my maternal line, I am 3/4 English, and this line immigrated in the mid-1800s. And then on that maternal line, the other 1/4 is Danish, and they also immigrated in the mid-1800s. So, fairly recent immigration, and we know exactly where they came from, 3/4 English, 1/4 Danish. Now, my paternal line, they are colonial ancestors who all came over 1700s early, and those lines are from the British Isles, Germany, France, and that is much more difficult to tease out exactly what belongs to which line. One thing that's interesting is my great-grandmother was full Danish, so I would have received roughly 12.5% of her DNA, but we know with random recombination, I probably didn't receive that exact number. So it'll be fun to explore and see what the companies report as we go through this episode.
And that's so fun to think about. What does your tree show? And then compare that with what the ethnicity estimates show. And also to remember about recombination, that you maybe didn't receive a full 12.5% from your great grandmother maybe only received like 8% from her. Who knows?
Right.
Well, let's talk first about 23andMe. Their report about ethnicity is called Ancestry Composition. They have a large reference data set, and they also use some public reference data sets such as the Human Genome Diversity Project. One thing that's great about the Ancestry Composition report is that it can break down the regions to specific areas. And I think they're one of the best for that because they break it down to smaller places instead of just saying 97% Europe. They break it down. So they break it down to like Northwestern Europe. And then underneath that, it has British and Irish, French and German, Scandinavian, and broadly Northwestern European. And then under Southern European, they have Spanish and Portuguese, broadly Southern European, broadly European, trace ancestry, and unassigned. So they have some things that are not that clear. But for the Scandinavian, for Diana's report, it says she has 7.9% Scandinavian. And then, it had the broadly Northwestern European, which is 18.1%. And so some of that maybe could be that Scandinavian as well. The 23andMe Ancestry Composition report also has like a few different things. It has a timeline, a parental inheritance chart, and a chromosome painting, which is hugely valuable. The timeline, it kind of estimates when different ancestors lived in different places. So it estimates that Diana's Scandinavian ancestors lived there in the mid 1800s, which supports, the documentary trail that we have. The other great thing about 23andMe is their ancestry composition chromosome painting. And you can see the segments which the company predicts originated from a specific ethnicity or region. So that's really exciting to be able to compare that with segments you've painted in DNA Painter that are from known matches and maybe assign a segment to a particular region or location. And then also possibly use that segment that is from a specific region to help you determine how that match is connected to you. So that can be exciting, especially if you can figure out if that segment is maternal or paternal.
Right. I love using the ethnicity estimate at 23andMe just because it lets you look at things differently. And I think that's so important in genealogy to make sure we're looking at things from all different angles. Well, let's turn to Ancestry DNA and talk about what they are doing. And so they have a DNA story and ethnicity estimate, and they have a reference panel made up of recently tested people with known roots in a specific region. They started this with a base of over 2,800 samples from 2 major genome projects, and then they added samples from their own proprietary reference collection and from DNA samples from customers consenting to participate in the research. So what Ancestry did was they first looked at family trees, and then they wanted to see if their lineages all traced back to the same geographic region. What I love about Ancestry is they give you a white paper that really explains how they did this, and they used quality control to remove outliers that might be introduced because of misattributions in the family trees. So they also treat some populations differently if they are thoroughly admixed, and then they only include individuals on the reference panel if they're part of a clearly delineated group. So how accurate are the percentages for each region listed on your DNA story? And I think we just have to remember the name estimates. And so as we click on each area, we see a range listed. And the percentage reported is the most likely, but Ancestry DNA explains that there are 1,000 reasonable estimates when compared to the reference panel. So for example, my current Ancestry DNA ethnicity estimate shows that I have 63% England, Wales, and Northwestern Europe, 12% Norway, which would be that Scandinavian part from Denmark, I am assuming, 10% Scotland, 6% Ireland, 4% Sweden, 3% Germanic Europe, and 2% Wales. So it's interesting that I have the 12% Norway, plus I have the 4% Sweden, which gives me more of a range of 16%. And this is so broad because some of that could come from my paternal side as well. It just shows me that, yes, I do have some Scandinavian ancestry from that full Danish great-grandmother, and so that makes sense. The regions displayed in your ancestry DNA story are based on your DNA, indicated with a solid circle. And then besides those, you have communities, which I think are so helpful. And the communities are going to evolve as the database grows. And I think we've seen that at one point we maybe didn't have communities, and then we logged on to Ancestry, and all of a sudden we have communities. And I know that happened with my DNA. My mother's DNA had a lot of communities, but I didn't have the same ones. And then gradually I'm getting more. So my mother's 25% Danish. And so she does have some good communities. She has some specific ones from England in the Midlands, Northwest England, Southeast England. And then she also has some specific Danish communities of Jutland, Zealand, Funen, Lolland, and Falster. Not sure how to say all those. But when I looked at the map, it was exactly right on with our known ancestry from Denmark. So that was really fun to compare that. And then she also has the community of Mountain West Mormon pioneers, which is where those Danish and English ancestors all came out to Utah. So it was really fun to see that her DNA ethnicity estimates was pretty accurate and clear, and they were just a little bit different than mine. It's always fun to compare you with ethnicity estimates of parents or siblings or children, and see how things are noted a little bit differently.
That is fun. After we wrote this book, the Ancestry, Ethnicity, Inheritance came out with the side view. So I was just looking at your side view to see, because you had wondered if the Norway and Sweden and Denmark, if any of that came from the paternal side. And what's interesting is that it thinks that some of it did come from the paternal side, but there's definitely larger chunks on the maternal. It actually has both for maternal and paternal. So maternal has Sweden and Denmark and Norway in larger amounts, and paternal has just smaller amounts of Sweden and Denmark and then Norway.
Right. And that's so interesting because I have no idea on my paternal side where that came from because they came over so early. So it's just fun to look at that. I love the new side view and the breakdown between paternal and maternal lines.
Right. And I just take it with a grain of salt too, because they just don't really know exactly where it came from. But it's cool to see the estimate.
Well, and we just have to remember that intermingling of populations. I know one of my friends is full German. Her parents both came over from Germany in the 1940s, early 1950s, completely German, and her ethnicity estimate shows all this different Scandinavian, things from all over Europe. So I think that just goes to show that our ancestors intermingled, and it's hard to just really figure out exactly if we have European ancestry where each little piece comes from.
Mm-hmm. Good reminder. Well, let's jump to family tree DNA. Their ethnicity estimate is called My Origins, and they define 24 population clusters that include ancient origins, as well as their unique population reference set. They use several projects to make up the reference set: the Human Genome Diversity Project, the International HapMap Project, the Estonian Biocenter, and the 1000 Genome. So they've validated these projects' data against individuals in the gene by gene DNA database who have proven ancestors in a specific area. So very similar to what Ancestry does. When you click on these different population clusters, the Learning Center opens up and describes about it and tells about the ancient origins of those people. And looking into these population clusters can help you learn about the diverse groups of people that inhabited a region. So that can be helpful and interesting. With Diana's DNA, it reported her ethnicity as 100% European and a breakdown of 55% Central Europe, 40% Ireland, and 5% Scandinavia, with less than 1% Finland, which I think is a new area for you.
Yeah.
So, and it's interesting that it only reported 5% of Danish, and the rest of that probably is included in the Central Europe region. So, this is an example of how the results are more accurate to the continental level.
And just again, how each region is defined by the company. they just have different ways of talking about it. They don't even discuss British Isles there. It's just Central Europe or Ireland. And so I think, oh, my British Isles is lumped together into Ireland, which is 40%. So it is kind of fun just to look at the different companies.
Yeah.
Well, let's talk about living DNA, which I think is so interesting. And living DNA just defines their ethnicity estimates as ancestry. But they have a unique algorithm that shows more regional details for the British Isles, and it breaks Great Britain and Ireland into much smaller regions. So if you have ancestry in the British Isles, this gives you some really great potential for new discoveries. And because I have so much British ancestry, it was really good for me to look at. So I get to see that I have some very specific regions noted. So for instance, I have 14.6% reporting of Southeast England, and that corresponds perfectly with my great-grandfather's line out of Dover in Kent County. And then my Danish ancestry appears as part of the Scandinavia region. So I think that living DNA can really help to pinpoint some specific areas within the British Isles. So a couple of the other regions that they defined for me, for my Great Britain and Irish ancestors, they defined the Southeast England, a little bit out of South Central England, South Yorkshire, Northwest England, Northumbria, Central England, East Anglia, South Wales border, Ireland, Cumbria, and North Wales. And some of those are very small, like 1.6%. But I think it's really interesting that they can take all my English ancestry and break it down so small. And I will be curious to see if that changes through the years as they increase their reference population and as the science gets more specific. But if you have got, British ancestry, how helpful would that be? That's really awesome. So they also have to note that if you test with living DNA, you'll get a maternal and a paternal haplogroup. It won't be as specific as what you get on family tree DNA, but it will give you your basic haplogroup.
Yeah, testing at family tree DNA with the Y-DNA or mitochondrial gives you like a very specific haplogroup, especially if you do the big Y700. Otherwise, the Y-DNA just gives you an estimate, like a high-level haplogroup. But I didn't know earlier when we were talking about 23andMe that they also provide a maternal and paternal haplogroup, if applicable, with their autosomal test results.
Right, and that could be really helpful in comparing to another DNA match to just take a look and see if you have the same haplogroups, and that can help you to see, which lines you might be sharing on. So that's kind of fun. With living DNA, I had my son test there, and so we do have the haplogroup, and it shows our population out of the Sami group, which is up Norway, Finland, way up north, which, yeah, that corresponds to that maternal line, which goes straight back through my Danish great-grandmother. So it made sense.
Let's take a minute and hear from our sponsor, newspapers.com. Did your ancestor disappear from vital records? Maybe they moved or got married. Newspapers.com can help you find them and tell their stories. Or have you ever had trouble figuring out how people tie into your family tree? Newspapers are filled with birth notices, marriage announcements, and obituaries. Items like these are a great resource for determining family relationships. On newspapers.com, you can explore more than 800 million newspaper pages from across the US, UK, Canada, and beyond in just seconds. Their easy-to-use search feature lets you filter your results by date, location, a specific paper, and more. When you find something interesting, the newspapers.com clipping tool makes it a snap to share it with family and friends. You can even save it directly to your ancestry tree. For listeners of this podcast, newspapers.com is offering new subscribers 20% off a Publisher Extra subscription, so you can start exploring today. Just use the code FamilyLocket at checkout. Let's talk about MyHeritage and their ethnicity estimate. They have included genetic groups that identify ancestral origins among 2,114 geographic regions. And the data set for genetic groups comes from the DNA data and associated family trees contributed by millions of worldwide users. Looking at Diana's results at MyHeritage, she had mainly Northwest Europe and England percentages, with 2 additional regions not reported by the other testing companies, East Europe and Iberian. So that was interesting. The North and West European regions are likely where the Danish heritage was included, so it wasn't broken down to Danish. But viewed on the continental level, the origins of Diana continue to be broadly European, just reported a little bit differently here. And once again, we see that little piece of Finnish DNA. So that was interesting.
Yeah, I'm so curious about the Iberian and the East European, where that comes into play, because that's really not anywhere else. So fun. Well, let's finish up this section on ethnicity estimates with the companies talking about GEDmatch. And maybe you've never used the GEDmatch tools, but they have a tool called Admixture Utilities. So you'll see they use the term admixture instead of ethnicity estimates, and they give you different ways to look at your origins. Several third-party developers have created calculators to compare your DNA to a specific reference population. So you get to choose. Do you want to compare it to European, African, Eurasian, etc.? And each calculator page provides information about the developers and links to their website. So to experiment with this, you first select Admixture from the list of DNA applications, and then you select a project from the drop-down menu, and then how you want to process it. And then you enter your kit number and select the calculator model to use. And then the program will generate the charts or graphs comparing your DNA to the reference population that you selected. So because I have a European heritage, I selected the Eurogenes K13 model, and it generated a list of percentages and a pie chart, which was fun. I liked seeing it visually a little bit differently. And it showed a large region labeled North Atlantic, which reflected my English and Danish ancestry. And it was interesting because this did show a little bit of Mediterranean ancestry, just like my heritage showed some Iberian and Southern Europe. So this GEDmatch Eurogenes K13 model showed that I had 9.81% West Mediterranean, 5.7% West Asian, 2.6% East Mediterranean, 1% Red Sea, which I found very fascinating. So if you have not looked at GEDmatch, go take a look. It's kind of fun to see what it will show you, and it also will show you broken down by chromosomes, the different regions. And I found that it actually correlated pretty well with how 23andMe breaks that down by chromosome. So fun to compare across the different companies.
Neat. Well, that's all the different companies. Now let's just talk briefly about the limitations of these ethnicity estimates. So they can be very useful, but there are limitations, and it's good to remember that as the science develops and the reference populations grow, your ethnicity estimate will likely change. And we have to also remember that some origins are difficult to accurately identify using DNA because of the broad migration between countries. So, looking at your ethnicity estimate on the continental level is usually going to be the most reliable way, rather than pinpointing specific countries of origin. And while that's exciting to see those specific countries, they could be inaccurate. So, just be aware of the potential for change and for inaccuracies. And also, we need to be wary of the potential for us having confirmation bias when we're looking at our ethnicity estimates. We might be looking for a specific hoped for ethnicity, and then when we see it, we're like, yeah, that's definitely correct. But we need to just be objective when we're evaluating our results and not search for what we hope we'll see.
We also don't want to get too worried if we see something pop up that looks unusual, just like that Western Mediterranean. Just know things will change, and the science will get us more focused on figuring out the correct ancestry as we go. But also, you might have a huge clue if you have a large amount of an unknown ethnicity that will tell you maybe something's incorrect in your family tree. So we also have to be aware that might happen. And we have seen that in our client work. So let's talk about locality research. We spent a lot of time on ethnicity estimates. And when we are using DNA with documentary genealogical research, we are looking for two people living in the same location where conception could have occurred. And how do you determine that location? Well, after we analyze the DNA and existing records, we probably have a specific research locality that has emerged. And this is where we're going to search for new documentation, more genealogy records that will help us to confirm or reject a hypothesis. And to create our viable research plan, we've got to study the locality. We teach to create a locality guide that helps us understand the history, laws, geography, and records that inform the research. And some of the things we can include in a locality guide are gazetteers, migration routes, jurisdictions, and boundary changes. It's so important to do the study of the locality and to study the locality as much as we are studying the records. The reason we haven't found the answer yet is because we don't understand enough about where the records are or why they were created or what was happening in the area where our ancestor lived. And so we recommend you do a locality guide, and it can be on any level that you feel will help you. Generally I like to go local with a county guide, but it could be very beneficial to do a state locality guide to understand records on that jurisdiction, or even on a country level if you're new to researching in, say, Ireland or Denmark or Germany, just to understand more about how record keeping in that specific location. So remember, your locality guide is fluid. You get started with the bones of it, and then you can add to it as you need. But as you get started, just keep it simple and focused on the time and the specific area that you think you'll be researching in.
Great. Well, to learn more about locality research, make sure you go to our chapter in our first book, Research Like a Pro, about locality guides. And there's a robust section just talking about how to create those and some examples in the work samples section. Well, now let's just talk about 3 case studies where either ethnicity, location, or both were important in discovering the genetic relationships. So the first one is an adoptee case where we were trying to find the biological father, and the birth mother was located early on, but the birth mother didn't tell anything about the birth father, so DNA testing was used. And the first thing that was noticed is that the test taker had a sizable amount of Finnish in her ethnicity estimate, indicating she was probably about 1/4 Finnish. Subsequent DNA matches that were found on that one side of the family that we believe to be the father's side, some of them had this piece of Finnish ancestry. And so you can see at Ancestry DNA the ethnicity estimate for your matches. And looking at that can really help you narrow down which line they're related to you on. And so that's kind of what was done with some of these different DNA matches who had, high percentages, like 12% of Finnish admixture, and then eliminating other DNA matches who shared with other people who didn't have the Finnish DNA. They were able to separate into 2 distinct groups. And then using surnames and locations from their family trees, they were able to narrow it down to a specific family and then find all the descendants of those people and then find a specific paternal group that had localities in Montana where the conception likely occurred. So that was exciting to be able to use ethnicity and locality to be able to solve this case.
That was really a fun case. And one of the interesting things about that was that there was this little bit of Native American ancestry in a couple of the DNA matches that weren't in the client DNA. And so, that threw in another little wrench, like, where does this come in? So I love looking at locality and ethnicity estimates as we're working through a DNA case. Another case that we worked on was uncovering an unknown grandfather. And in this case, we have Jack Jr., who never knew his biological grandfather. And as a young boy, his father, Jack Sr., had just found his birth certificate, and his surname was Green, which was not the surname of his adopted father that he had used his entire life. And his mother snatched that certificate away and said, "We're never talking of this again." And he always wondered about this mysterious name of Green. So he turned to DNA testing after his grandmother and father's passing and took the Y-DNA test as well as the autosomal DNA test at each of the DNA testing companies. And it revealed the surname of Robb, which was not Mr. Green. It was a completely different surname. And so correlating that and some autosomal matches found a likely candidate for his grandfather. So then we had to turn to locality, trying to figure out a place where this man and Jack Sr.'s mother had been. And so it turned out, using all the documentary research and studying the locality, that they were both in Oklahoma City, Oklahoma in the 1920s. And so it became evident that yes, this could have been the father, and the mother and the father were in the same area within a few blocks of each other at the time of conception of this child. So we really had to use location in conjunction with the DNA to solve this case. And this is how this always is with our DNA research. We can't just do DNA alone. We always have to turn to locality next and put together the pieces of the puzzle.
All right, our last case study is about connecting a female ancestor to her father. And as we know, female ancestors born before 1850 are often a brick wall for us. And with Rachel Cox, she was no different. Her marriage record revealed the surname of Cox, and she was married in Navarro County, Texas, to Hickman Monroe Schultz, but no father was listed on that marriage record. But research eliminated all Cox men in Navarro County in the 1840s except for one named Benjamin Cox. He was listed in proximity to Rachel's husband on a list of road commissioners, so he seemed like a good candidate. And researching him uncovered a migration path from Ohio to Indiana, Arkansas, and Texas. So creating locality guides for the counties in these areas that he lived in and understanding the available records there made it possible to research and understand Benjamin Cox's localities in that time period of migration. And some overlapping localities continued to point to a relationship between Rachel and Benjamin. Next, DNA was used to see if the hypothesis would be supported or rejected with additional evidence. And so adding Benjamin Cox as the father for Rachel on our ancestry tree caused through lines to be generated to Benjamin Cox. And there were over 70 possible DNA matches to Diana as a direct descendant of Rachel Cox to descendants of Benjamin Cox. And viewing the localities of these associated family trees again revealed these locations of Ohio, Indiana, Arkansas, and Texas, correlating with the records for Rachel and Benjamin. So, after much analysis and inclusion of additional DNA test takers, the DNA evidence did provide final confirmation of what the records indirectly showed. So it was exciting to see how correlation of different locations in a migration path was able to confirm the identity of Rachel Cox's father.
Right, that was such a great brick wall case to solve finally with DNA. Well, let's give you a task. So, the task for this chapter is to explore ethnicity estimates and locations of shared matches. So first of all, consider what role ethnicity estimates could play in your research project. View the reports on each of the company websites to see if they match your genealogical family tree, and make notes about any conclusions in your research project document. And then compare your DNA matches ethnicity estimates to yours. Do they provide clues to the research question? What localities are named in your DNA match trees or on the company websites? And add this information to your research log. And then second, you're going to create a locality guide. So now you've created a research objective and analyzed the evidence, so you can create a locality guide or guides to inform your research. Look at the DNA matches that might be related to you on the same site as the subject of your objective. Determine the time and place where their ancestors lived. Do they seem to match with what you know? If so, this will give you confidence in following this line of reasoning. And then you will review your timeline analysis and decide which localities seem reliable, which may hold the answer to your research question, and then you'll choose your time and your place to focus your locality guide. Keep in mind you may need to do 2 or 3 smaller guides if your ancestor migrated, such as the Benjamin Cox project, where I had to do some locality guides in Ohio and Indiana and Arkansas and Texas because he was a migrating ancestor. But in contrast, the Oklahoma City project only needed 1 locality guide because the research centered right there in Oklahoma City. So just know every research project is unique, but Every project will need a locality guide to help you better understand your ancestors' time and place. It really is the key to your research planning and finding the answers that you've been looking for so long.
Well, good luck with your task. And next week, we will talk to you about Chapter 8, which is about DNA tools and methodology.
All right. Bye-bye, everyone.
Bye. Thank you for listening. We hope that something you heard today will help you make progress in your research. If you want to learn more, purchase our books, Research Like a Pro and Research Like a Pro with DNA on Amazon.com and other booksellers. You can also register for our online courses or study groups of the same names. Learn more at FamilyLocket.com/services. To share your progress and ask questions, join our private Facebook group by sending us your book receipt or joining our courses. To get updates in your email inbox each Monday, subscribe to our newsletter at FamilyLocket.com/newsletter. Please subscribe, rate, and review our podcast. We read each review and are so thankful for them. We hope you'll start now to research like a pro.